and begin with the first objective. Describe the various properties of graded potentials, including the direction of change in a potential, the magnitude of change, temporal and spatial summation. We'll talk about what those two features are. And then lastly, explain how graded potentials in neurons can trigger an action potential. So to begin this discussion, we want to think about the resting membrane potential, because starting off at that -70 is the baseline to then create an action potential. So we want to think about our neuron hanging out at rest, not excited or activated. And if you were to put a voltmeter into that neuron, the reading that you would get is -70 millivolts. And what that means is that there's an abundance of negative charge on the inside compared to the outside, which is more positive. So on that basis, we can then begin to talk about different types of signals that can begin to manipulate this -70 and can change the membrane potential. So we'll talk about different types of electrical signals, and those can be small, short communications or short signals, which are called graded potentials, or they can be larger, more meaningful, more functional signals, electrical signals, which are called action potentials. And those will travel for longer distances, longer periods. So let's look at the graded potential first. So if we think about the two images on the right here, and we're comparing the membrane potential to the time, we know that we start off at -70. So though it's not shown here, the resting membrane potential, the baseline for change in the membrane potential really begins at -70. And then we can have small blips, right? Small increases in the membrane potential that are not necessarily strong enough to be sustained or to travel along the length of the neuron, for example, but they do change the membrane potential for a short period of time, and they are small enough changes that they could potentially sum together to create a larger, more impactful change. So that would be the graded potential. And notice how the membrane potential changes very temporarily for a short period of time, and then it comes back down to rest. Now we can have positive changes, which we'll talk about what those are coming up, or we can have negative changes, which is where the membrane potential dips even more negative than -70, and then it comes right back to -70. So these smaller changes that are short in distance, short in time, not sustained, but can eventually be added together or aggregated to generate a larger change, those would be your graded potentials. Alternatively, the action potentials are larger. They communicate over longer distances in time, and these potentials have the ability to travel the length of the neuron and then eventually signal the adjacent neurons that are synapsing onto this neuron. So they have the ability to actually send the signal to an opposing cell. And these action potentials are going to be the result of smaller graded potentials that have been added over time, or that are close enough in space that they can also be added or summed. So on the right here, again, we start off at -70. Here's a small graded potential. Here's another graded potential that's a little bit larger, still not large enough on its own. And then that is added with an even larger graded potential that gets us to -55. And the reason -55 is so important is because that is what's called the threshold value. When graded potentials are strong enough to get to -55, then we trigger an action potential. And so this large spike in the membrane potential that goes all the way up to about +30 or so, and then comes back down below -70, and then gets back to -70, that large change in the membrane potential, that spike, is what we refer to as the action potential, okay? And again, that's more meaningful, that can travel longer distances and signal the adjacent cells. So let's talk a little bit more about the neuron, where along the length of the neuron this is happening, and the different types of potentials that we can see. All right, so let's look at the potentials. And we've used the word potential quite a bit so far. And simply put, a potential is just the ability to do work, the ability to send a signal, the ability to communicate, right? And so the energy that is created by the gradients, by the channels that are opening, these create these potentials, which are signals that have the ability to communicate. So let's look at a neuron and let's kind of describe the parts of the neuron while we're at it. So here's our soma. Right? Here's our axon. And then here's our axon terminal. Okay. And then of course we have a little nucleus somewhere in here. Right, little nucleus. So this is the soma, also referred to as the cell body. These are synonymous terms. So soma or cell body. This is the axon, which is the projection towards the axon terminal. Okay, and now potentially there's going to be either another neuron here or a muscle cell here. So the axon terminal is going to be synapsing onto some other type of tissue. Again, either another neuron, a muscle cell, a cardiac cell, one of the other excitable cells will be synapsed. So the soma is going to be the proximal part of the neuron. And we also have axon terminals from other neurons that are synapsing here as well. So here's an example of another axon terminal from an adjacent neuron that would be synapsing onto this neuron as well, okay? So we want to think about this as a chain of communication between neurons to neurons. Now, in this example, this would be the presynaptic neuron because it is before the synapse. So this is an example of a presynaptic neuron, whereas the neuron that we're focusing on would be an example of a postsynaptic neuron. As a result, this area adjacent to both structures would be a synapse, right? This area of communication would be a synapse. All right, which is kind of where all the magic happens as far as transferring of the signals. And so each of these would have their own area of synapse. The other important parts of the neuron we want to describe is the axon hillock. So the axon hillock is a region near the narrowing of the neuron. I like to call this the neck of the neuron. So it's where the soma becomes the axon. And so this is called the axon hillock. And this has a lot of importance as far as this is where graded potentials are added together, and then this is where action potentials are actually generated. So if we divide our neuron into different parts down here, this would be sort of the threshold region to decide whether or not we fire an action potential. So proximal to the axon hillock, we have lots of graded potentials that are coming in from signals from the presynaptic neurons. So before the axon hillock would be graded potentials. And then after the axon hillock is where we add all of those graded potentials. They are summed at the axon hillock. And then this is the region where we determine if they are strong enough to meet that -55 threshold. If so, beyond the axon hillock, we will trigger the action potential. all right? And that is going to move all the way down towards the axon terminal, okay? Let's talk about some of the different types of channels that we have throughout the neuron. So on the soma or cell body, we have these projections that are sticking out. These are called dendrites. These are called dendrites. And so along the soma, along the dendrites, what we have are ligand-gated channels. Right, I'm going to put that in brackets here. So these would be ligand-gated channels. Okay. And ligand-gated channels, we described this earlier. This just means that something has to bind to this channel in order for it to open up, whether that be a particular neurotransmitter or a particular ion. Something has to bind to this channel, and the gating mechanism on this channel requires a ligand to bind. Along the axon hillock, we have voltage-gated channels. Voltage-gated channels. So the gating mechanism here is a little different. So voltage-gated channels, right? So here, the channels will open up depending on the membrane potential. When the membrane potential gets to a certain value, then it's going to trigger the opening up of voltage-gated channels. And that begins to make sense, right? Because we said that the axon hillock is the area where we sum the graded potentials, determine if we reach that threshold value, and then if we reach that threshold value, that would trigger the opening of these voltage-gated channels. And then the action potential would propagate down the length of the axon, okay? Along the axon, we're also gonna have voltage-gated channels, so that would kind of match what we just described. Voltage-gated channels. All right. And then we have at the axon terminal, we're going to have calcium channels. And calcium channels are going to be important because they're going to trigger exocytosis. We talked about exocytosis, which is the dumping of the secretory vesicles into the synaptic space, and that requires calcium. So at the axon terminal, we've got calcium channels. Okay. Now, I'm going to just mention it here just so that we can bear that in mind. The leak channels that we've talked about, right, the ones for potassium and sodium, the ones where potassium has many more of those channels, those are referred to as leak channels. And those are going to be all throughout. So leak channels for sodium and potassium are all throughout the neuron. Okay. I'm also going to put here the sodium potassium pump is also all throughout the neuron. Okay, so while we haven't drawn these in, because they're not necessarily concentrated or focused in one area, we want to think about them as always being on the cell, right? On the basolateral surface, we have the sodium potassium pump, and then we have the leak channels all throughout as well, where sodium can leak in, potassium can leak out, and we have a higher permeability. Remember, we have a higher permeability for potassium and a lower permeability for sodium. Okay. All right. Let's make sure I didn't forget anything. I'm going to put here as well. So we have ligand-gated channels on the dendrites as well as the soma and the cell body. So I'm just going to repeat that here so it's clear. Ligand-gated channels here as well. And then I'm going to make a note of this threshold value here at the axon hillock. So the negative 55 millivolts, which is the threshold, is decided at the axon hillock. So, oops, I missed the E here. That's going to be important for this region.